Hello and welcome back to Excel Share Academy, guys. And today we are going to learn about the DNA polymerase and how the processivity of the enzymes, like how fast the enzyme can add or catalyze a reaction. Okay. Before that, so in the previous lecture we talked about the role of the metal ions, right? If you remember, we talked about that two divalent metal ions are required, right? Now, in the previous lecture, I forgot to mention, we have seen that either Mg2 plus can be used and zinc 2 plus can be used. It was mentioned, right? In many cases, you will see that Mg2 plus is being used mainly cofactor की तरह. Zinc ज़्यादा use नहीं होता है. कोई एक reason दे सकते हो why not zinc? What you know about magnesium and zinc? क्या रीजन हो सकता है जिसके वजह से the enzymes mainly use magnesium as compared to the zinc?
Is it availability?
No, it's not because of the availability. Like if you're there in the lab you have got plenty of magnesium chloride and zinc chloride. Why do you think enzymes will prefer magnesium more as compared to the zinc? थोड़ा सा और सोचो. क्या लगता है? What do you know about magnesium and zinc? अच्छा, I'll give you another hint. Mg2 plus and zinc 2 plus. Huh? What about size? मैंने एक हिंट दिया है, क्या लगता है? ओके, एटॉमिक नंबर ऑफ मैग्नीशियम कितना है, कोई बता सकता है? Anyone? See, this is what I was talking about. It's very basic, right? So you have got 12. Zinc का कितना है? Around 30. ठीक है. With this much information, what do you think, guys? Which divalent metal ion is having bigger size? Even though both are 2 plus, which one is going to have maximum size?
Zinc.
So that much size, if suppose it is taking over here, right? So what will happen? It is, and that too two we need over here. So just imagine the scenario where the zinc is occupying the maximum space. यहाँ पे 12, यहाँ पे 30 है. It is like double, more than the double the size, right? So you can see that it is going to occupy maximum space, because of which the alignment of your triphosphate and the attack of 3-OH on the alpha phosphate, that won't be in the proper alignment. Okay. ये काफी टेक्स्ट बुक्स में नहीं दिया है, but once you refer more textbooks, like when you go higher studies and all, you'll come to know about this. So as I mentioned before, you start your basics with Watson textbook, like from there this figure also have taken, and the next book which I have always mentioned is Robert Weaver. Okay. Now, if you could complete Watson and Robert Weaver, then you can proudly say that you have completed or you are well thorough in mole bio concepts. Then nobody can beat you. Acha, tell me, this much concept is clear now? Like why zinc is not being used maximum by the enzymes and then magnesium is used. Later on, you will also see that, if suppose, you will also see that if I use 50, sorry, if I use 50 millimolar of magnesium chloride and 50 millimolar of zinc chloride. Okay, so what you see over here is that the concentration or the molarity of both the salt is same. Still, magnesium chloride gives you better activity of the enzyme as compared to the zinc. And you will also see that zinc is resulting in the precipitation of the enzyme. Okay, now what do you mean by precipitation? It is enzyme precipitation means same thing with the protein precipitation, right? Now, whenever protein gets misfolded, Okay, they tend to aggregate with each other. So many a times you see that zinc ho gaya, aapka cadmium ho gaya. So all these salts are metal ions are going to help in the protein precipitation or enzyme precipitation. Maximum times you will see that manganese, magnesium are good. They actually help in the establishing a interaction between the substrate and the enzymes. ठीक है, ये हमेशा याद रखना इतना. Okay. Now, the PAM domain also monitors the base pairing of the most recently added nucleotides. Now, what PAM domain? So, we were talking that in the previous lecture that our DNA polymerase, it looks like your right hand, right? So, fingers ho gaye, thumb ho gaya, and then you have got the palm region. So, this PAM domain, okay, so this PAM domain, it is also helping in the base pairing. Now, this region of the polymerase makes extensive hydrogen bond contacts with the base pairs in the minor groove of the newly synthesized DNA. So, what is happening? जैसे जैसे आपका DNA replicate होता जा रहा है, यानी कि synthesize हो रहा है. So, what is happening? The minor groove of the DNA, if you remember the structure, the minor groove of the DNA are making base pairs with the newly synthesized. The minor groove is making with the polymerase का palm region. ठीक है. Now ये भी किस लिए? Because it needs to have a proper helical structure. Otherwise, what will happen? The DNA is losing its property. Okay. So these contacts are not base specific. यहाँ पे हमें किसी sequence की जरूरत नहीं है. It can be randomly. Okay. But only form if the recently added nucleotides are correctly base paired. So mismatched DNA in this region interferes with these minor groove contacts and dramatically slows catalysis. Now how it is going to slow down the activity, as I mentioned before, अगर आपने ध्यान दिया हो, so we have got the like this, and उसमें अगर आपका mismatch आ गया है, so it will look like this, एक bend आ जाएगा. So this is going to interfere with the movement of the DNA polymerase ahead, and this will slow down the activity of your DNA polymerase. Okay. The combination of this slowed catalysis and reduced affinity for newly synthesized mismatched DNA, it will allow the release of the primer strand from the polymerase active site. अब क्या होगा? So finally, the primer will get detached from the template and your DNA replication will stop there. क्यों? क्योंकि वहां पे अभी हमेशा हमें repair करना है because there was a mismatch detected. Is this much clear? This slide is clear to everyone?
Yes, ma'am.
Okay. Now the fingers. So अभी हमने जो discuss किया था, it was all about the palm region. Now the fingers are also important. So several residues located within the fingers, they bind to the incoming DNTPs. Incoming DNTP. So हमेशा याद रखना the function of these. So एक हो गया अभी. Just now we have discussed about the palm domain. Now we are discussing about the fingers. So इनका काम क्या है? जो भी incoming आपका DNTP है, sorry ऐसे लिखते हैं, to interact with that. So once it is correct one, then only it is going to make the interactions with that. Okay. Now, more importantly, once a correct base pair is formed between the incoming DNTP and the template, the finger domain moves enclose the DNTP. अभी कैसे है? Just imagine that the fingers are making interactions with the DNTP. Now it is almost closing the fingers and bringing towards the domain. Imagine हो रहा है क्या आपको? एक second. Yeah. Maybe I can show you like this. See, if you can have like this, okay? ये आपका fingers है, ये हो गया आपका thumb, ये palm region जहाँ पे आपका DNA synthesis होता है, okay? So what is happening now is that जैसे ही इस region में DNTP आ रहा है, it is making a correct interactions, right? Then once it is a correct DNTP, it is going to move it like this, bringing towards near the palm region and makes it easy for the catalysis to occur. ठीक है? So this closed form of the polymerase hand stimulates catalysis by moving the incoming nucleotide into close contact with the catalytic metal ions. Now, look at the structure. This figure, not the structure. So this is really very important because बहुत बार आ चुका है, and I'll tell you what questions they have asked. So two types of questions were asked in this. Pehla, by what angle it is present, and what are the amino acids which is responsible for this detection. Okay. So first one. So this is a figure where you can see the interactions of the fingers. So just imagine ये यहाँ पे fingers से आपके, ठीक है, आपके DNA polymerase के. So the change is a 40-degree rotation of one of the helices in the finger domain called the O helix. Okay. So यहाँ पे 40-degree change हो रहा है. See, I told you, right? It is bringing towards the palm region. So कितना move हुआ है आपका? 40-degree move हुआ है. Okay. Now, what are the interactions which is forming? It is between the tyrosine, lysine and arginine. Okay. Now, यहाँ पे भी important है. Why do we have two positively charged amino acid and one is tyrosine? Tyrosine क्या है आपका? Aromatic amino acid, right? Now look at the significance over here. So why these three only? So we know that lysine and arginine, they are positively charged. Now, why do we need positively charged amino acid in order to interact with the incoming DNTP? What will be your answer? Answer is very straightforward. So yes, what will be the answer?
DNTPs are negatively charged.
Yes, very good. So they are negatively charged. So before, like in the previous lecture, we discussed that how the two divalent metal ions were helping in stabilizing the structure, right, in the catalytic site of the DNA polymerase. Now, same thing. Again, it is also again just to make sure that that is the correct DNTP and it is stabilizing. So you can see that here. अभी देखो दूसरे एंगल से देखो. So in the previous we saw that metal ions are making interactions with the DNTP. Okay, 1, 2, 3 phosphates are there. So you can see alpha here, beta and gamma phosphates are there. You can see that your first metal ion, second metal ion, they are making the interactions. Now on the other hand, when you see, what do we see over there? Sorry. That lysine and arginine is also stabilizing the phosphate. Now, why so much stabilization is required? Because if you see the structure, it is something like this, right? Oxygen negative, oxygen here, here negative, right? So this is how. So यहाँ पे अगर दो तरफ से अगर आपको metal ion stabilize कर रहे हैं तो दूसरी तरफ से यहाँ पे we have got lysine and arginine stabilizing the incoming DNTP. Now, third thing, what is happening? Now your tyrosine, it is making a stacking interactions with the nucleotide, with the bases. जरूरी नहीं है वहां पे GC होना चाहिए. It can be AT also. यहाँ पे A हो सकता है, यहाँ पे T हो सकता है, यहाँ C हो सकता है, यहाँ G हो सकता है. ठीक है? So it is making the stacking interactions with the base of the incoming DNTP. Okay. So overall, what is happening now? You can see that the fingers are playing so important role. First, it is stabilizing the incoming DNTP by using lysine and arginine in stabilizing the beta and gamma phosphate. Okay, along with the divalent metal ions. Secondly, tyrosine is making stacking interactions with the base of the newly incoming DNTP. So all these combination of these interactions, okay, they are positioning the DNTP ready for the catalysis to be done by the DNA polymerase. Yes. Now tell me, is it clear to everyone? Akshaya, is it clear? Can you repeat?
Yes.
जो भी अभी मैंने बोला है?
Yes, ma'am. So there are 3 amino acids which stabilize the incoming DNTPs. So arginine and lysine are positively charged. So they stabilize the incoming DNTP. Even though there are two the metal ions which are stabilizing on one side, these two also make significant stabilization on the other side. And tyrosine makes the stacking interaction on the base of the DNTPs. So So totally this is stabilized, and there is a, a, a rotation of 40 degrees on the helices on the finger domain. So they are known as O helix.
हाँ,
Yes,
अक्षय
ma'am.
सब बोला, सही बोला, थोड़ा सा ऑर्डर गलत हो गया. फर्स्ट इट इज द रोटेशन ऑफ द ओ हेलिक्स and the interactions that are happening. See ना यहाँ पे, so you can see that this is the O helix, the fingers का एक O helix है. Now, when this rotation is happening over here 40 degree, then only these interactions with the basic amino acids as well as the tyrosine it is happening. Okay, rest all is correct. The order थोड़ा सा गड़बड़ा गया था. Okay, so I believe that everyone understood this thing. Yeah. Now, question इसमें से क्या क्या आया था? As I mentioned already about it is the 40 degree rotation. ठीक है, एक ये आया था, and the number sorry, the name of the amino acids which were required for the stacking interactions. So obviously confuse करने के लिए we have 3 aromatic amino acids: phenyl, tryptophan and tyrosine. और ऐसी fourth होगा कुछ, none of the above or all of the above, I don't know. So this is how you need to remember. Okay. तो stacking interactions के लिए tyrosine is there in the DNA polymerase fingers. Okay. Now next topic. So DNA polymerases, they are the processive enzymes. Before I proceed, one second. Okay, so DNA polymerases, they are the processive enzymes. Now, when we talk about the processivity of the enzyme, what does it mean? हम कहना क्या चाहते हैं about the enzyme? We are talking that how fast an enzyme can catalyze a reaction. ठीक है? Okay, so let me ask basic questions. How much time does a cell takes to divide? Let's talk about eukaryotic cell. So how much time does a cell take to divide? Yes. No one? Tanvi? Yes, Tanvi? ऐसे ब्लैंक मत हो जा, मुझे से लगता है जैसे मैं डिस्कनेक्ट हो गई हूँ. हाँ. अक्षय, श्रुति, you can also give the answer. ये बहुत बेसिक चीज है, ये आपको मालूम होना चाहिए. Yes.
24 hours, ma'am.
Yes, 24 hours. And when you say 24 hours, इसमें if you know about the cell cycle phases, how much time does mitosis take and how much time the synthesis of DNA takes? Any idea?
Ma'am, mitosis takes 2 hours.
Okay, let us say that mitosis is taking 2 hours. So 22 hours में फिर आपका G1 phase है, S phase है, G2 phase है, right? थोड़ा बहुत तो आपको मालूम होगा सेल साइकिल के बारे में, right? So basically it is not 2 but 23 plus 1 we say. ठीक है? So 23 is your interphase and 1 is your mitosis, M phase जो हम कहते हैं. So just imagine, even though we are saying that DNA polymerase is rapid, but still it is taking 23 hours. Okay, of which DNA synthesis is the maximum. Now you tell me guys whether it's a slow process or it's a fast process. अगर मैं ये बोल रही हूँ 23 hours में आपका DNA synthesis होता है, ठीक है? If I say that our DNA polymerase is slow, whether I am wrong or right, justify, or if I say fast, then again justify. See, these are the questions which comes for your interview. जब भी आप कहीं पे जाते हो, तो आपको सोचना पड़ेगा. मेरा क्वेश्चन समझ में आया?
Yes, ma'am.
हाँ. Okay, let us say that Akshay is saying that DNA synthesis done by DNA polymerase is a slow process. Okay, Akshay ने बोला slow है, but Shruti is saying it is fast. Now, how you are going to justify yourself? Yes? Any comments? Should I pass?
Ma'am, I feel it is fast.
Okay. Can you
I feel
justify
as if
yours?
it's fast.
Okay. So everyone
Because
say, ha. Yeah. Yes. Yes. Sorry. Say.
DNA is too long and to synthesize these strands and all. I feel it. The work of DNA polymerase is very rapid. Like uh,
Yes, very good, very good. So the
Very
counterpoint,
efficient.
yes. So we know that DNA synthesis is very fast, as I already mentioned also here that it is adding in seconds. It is adding 1,000 nucleotides. It's actually very fast. Okay. Now, still it is taking 23 hours. इसका मतलब क्या? Because we have got such a long DNA. उसको इतनी जल्दी अब एक नहीं है हमारे पास. We have got how many? 23 pairs are there. राइट और उन सबको करना है सो इट विल टेक टाइम ओके सो दैट्स ट्रू सो डीएनए पॉलीमरेज जो है आपका बहुत ही एफिशिएंट है एंड सिंस आवर डीएनए इज सच ए लॉन्ग अगर ये इतना फास्ट नहीं करता सो इट वुड हैव टेकन मिलियंस इयर्स एंड हमारा कभी एजिंग ही नहीं होता लाइक एवोल्यूशन भी नहीं होता एजिंग भी नहीं होता जो पहले 100 जो हुए होंगे वही अभी भी 100 चल रहे होते हैं ठीक है सो इतना स्लो हो जाते हैं हम लोग anyways good so just have confidence okay whenever you give the answer so it's the correct answer now so the degree of processivity it is defined as the average number of nucleotides added each time the enzyme binds to a primer template junction okay so if I'll show you the video also in which we'll see how far the DNA polymerase is actually changing and adding the nucleotides now why it is changing if you know that in the case of the replication we have got leading as well as the lagging strand right so leading में तो एक DNA pol चलता रहता है मगर lagging में change होता रहता है we'll see how and we'll explain also why okay so each DNA polymerase, it has got the characteristic processivity that can range from only few nucleotides to more. So depending on the type of DNA polymerase, you can have the different processivity. For example, sorry. For example, in the case of prokaryotes, your DNA Pol 3 is more efficient as compared to 1. अब 1 का क्या काम है जहां पे भी रिपेयर होता है छोटा सा एक निक रहेगा या गैप रहेगा सो उसको फिल करता है आपका Pol 1 whereas Pol 3 is a main replicating enzyme. So obviously the degree of processivity of both Pol 3 and Pol 1 is going to vary. Okay. Yeah. So let's see over here. This is one example of that only. So look at the figure. So you can see over here. So you have got the template, green color का आपका primer है. So this is non-processive DNA polymerase. So यहाँ पे क्या होगा? One at a time आपका DNA polymerase add करेगा, फिर आगे बढ़ेगा, फिर धीरे-धीरे जाएगा, ठीक है? Whereas in the case of the processive enzyme, what it happens is one, it is going to add thousands of nucleotides together. And it is fast. So ये खत्म करके अपना निकल जाएगा. It will bind to the another strand and complete the DNA synthesis. Okay, so this is the just a representative way to show how a non-processive and a processive DNA polymerase will work upon. Now coming to the exonuclease proofreading activity. Okay, so now why we need exonuclease proofreading activity? Because of the tautomerism. Now what is tautomerism? Look at over here. So we have got cytosine which can base pairs with guanine, ठीक है? Keto guanine हो गया आपका ये. But if we have got the keto की जगह का enol guanine होता, so then instead of binding with cytosine, it will base pair with the thymine. So what will happen in the next round of replication? Instead of having CG in the strand, अगर suppose ऐसा मेरा strand है, यहाँ C है, यहाँ G है. Okay, so instead of this, in the next replication, what will happen? I'm going to have CG की जगह TG. तो नेक्स्ट में यहाँ पे TA हो जाएगा, यहाँ पे CG हो जाएगा, राइट? तो एक तो आपका पेरेंटल स्ट्रैंड ही हुआ CG एंड CG, बट जो दूसरा था वो आपका TA हो गया. सो व्हाट डज इट मीन? वन ऑफ द डॉटर DNA हैज गॉट रॉन्ग a base pairing, correct? जिसकी वजह से क्या होगा? This is going to result into the mutation, and that's why we need all this proofreading mechanism. Guys, this much is clear? ये clear है ये वाला concept? Yes, Shruti, is it clear?
Ma'am, your voice is breaking.
I couldn't hear anything up until now.
Yeah, अरे बोलना चाहिए ना. Okay, now it is clear.
Yes, ma'am.
Yes, ma'am.
Okay, so I'll just explain once again. Okay, so proofreading activity is required because of the tautomerism. Okay, tautomerism. Now, what is tautomerism? That your bases can exist into the keto form. Okay, keto form मतलब double O bond, or enol form where you have got OH group. ये हो गया आपका keto to enol form. For example, your guanine is basically a keto guanine which can base pair with cytosine. So you can see 1, 2, 3 base pair it can form. Okay, hydrogen bonds it can form. Now, if you have, instead of keto guanine, if you have enol guanine, what will happen? यहाँ पे double bond O के जगह you have got the OH. Okay, so again G की property क्या है? To form 3 hydrogen bonds. So instead of C, it is going to form 3 hydrogen bonds with T. Okay, because of which what will happen? In the DNA, instead of having CG, I am going to have TG, which is wrong, right? So next round of replication में I'm going to have TA and CG. In another replication, ये तो मुझे CG और CG ही देगा, but this is going to give me TA and TA. So these DNA is having or is carrying the mutation. ये clear है?
Yes, ma'am.
Yes, everyone, it's clear. So इसीलिए we need exonuclease activity or any kind of proofreading activity. Okay. Sorry. So these keto-enol formation of those bases results in the incorrect base pairing. So in order to correct this mistake, we have got the exonucleases activity. Okay. Now, exonuclease and endonuclease difference क्या है? Exo, this is from the DNA end. Endo, यानी कि ये बीच में से होगा. ठीक है? Within the strand, DNA strand बीच में कहीं पे ये कट कर सकता है. Exo means अगर यहाँ पे DNA है तो 3 dash से करेगा, अगर 5 dash है तो 5 dash से करेगा. Endo can come and cut from the between. इतना clear है everyone?
Yes, ma'am.
Okay. So proofreading exonucleases in action. तो अभी हमें मालूम है that your DNA polymerase have got exonuclease activity. ठीक है? तो वो proofreading कैसे करते हैं वो भी हम देखेंगे. So the removal of the mismatched nucleotide is done by the reducibility of DNA polymerase. Now क्यों? See, जब भी कोई भी mismatch होता है, ठीक है? Suppose ये हो गया and यहाँ पे कुछ गलत base pair आ गया है. Okay, so there will be a bend kind of thing, like which is not proper helical, but a bend is present over there. So because of which what will happen? ऐसे होगा, आपका base pairing ऐसे होना चाहिए, right? Instead of that, there will be a bend. तो जिसकी वजह से second nucleotide DNA polymerase is not able to do because of the previously wrong or mismatched base pair. So the second nucleotide होगा, incoming nucleotide होगा, it won't be able to sit properly into the palm region of the DNA polymerase. So then there won't be any 3-OH का nucleophilic attack on the alpha phosphate, ये सब नहीं होगा, जिसके वजह से your DNA polymerase has got the reducibility. So this will activate the exonuclease activity of your DNA polymerase. ठीक है, so polymerase as well as exonuclease activity, दोनों ही activity is present in the same enzyme. So this altered geometry, it reduces the rate of nucleotide addition in the same way that the addition of an incorrectly paired DNTP reduces the catalysis. Okay, so when a mismatched nucleotide is added, it both decreases the rate of new nucleotide addition and increases the rate of proofreading exonuclease activity. ठीक है, अब देखते हैं कैसे करता है ये. सपोज यहाँ पे देखते हैं, where you have got no DNA synthesis, so you have got the exonuclease active site is present over there. Can you see? This is your exonuclease activity is there. Okay, now look it over here. You have got the mismatch. Because of mismatch, the newly synthesized DNA is not able to form a proper helix over here, and there is a bend. Right, so this bend is pushed towards the exonuclease active site. So what is happening? The exonuclease active site is getting activated. Okay, because of the presence of the bend over there. Now, once the exonuclease activity is activated, it is going to remove the mismatched nucleotide. So what will happen? ये जितना wrong portion है, इसको आपने chop off कर दिया, हटा दिया. Again back to normal 3-OH. So once it is removed, again your exonuclease activity will go down and polymerase activity will come up. Okay, and the DNA polymerization will resume. Guys, this much is clear?
Yes.
Yeah, Tanvi, can you explain?
Yes, ma'am. So, ma'am, first, when the mispaired base pair, when it comes there, so so this mismatched pair, that bent goes to that exonuclease active site, and then it is removed, and then the DNA polymerase activity increases, and yes, it is then increased.
Okay, बहुत ज़्यादा short कर दिया था. But okay, I just want, see, in between I'm calling you guys because I want to know whether you guys are able to repeat the same or not. ठीक है, तभी समझ में आता है whether you have understood or not. Same way, see, again, it's not the bent of the mismatch is going to the exonuclease active site. It is because of the bent, the exonuclease site is getting activated. ठीक है, ये कहीं नहीं जा रहा है, ये palm region में ही है. Just because there is a bend, it is going outside of the palm region. What happens? That triggers the exonuclease active site. So ये आपका active हो जाता है. This gets activated. It is going to chop off the end of the 3-OH, removes the mismatch, and comes back to the normal. So once exonuclease has done its work, now the polymerase will resume its function. ठीक है? Same thing over here. So when a mismatched base pair is present in the polymerase active site, the primer template junction is destabilized. Okay. Now, the DNA polymerase active site binds such a mismatched template poorly, but the exonuclease active site has got the higher affinity. Obviously, right? जब आपका exonuclease active हुआ है, तो आपका polymerase की जो activity वो कम हो जाएगी, and vice versa. जैसे ही mismatch आपने remove कर दिया है, exonuclease activity कम हो जाएगी and polymerase activity high हो जाएगी. Thus, the newly unpaired 3-end moves from the polymerase active site to the exonuclease active site. Exonuclease अपना काम करेगा. It is going to remove the mismatch, right? And then again, polymerase active will be resumed. So, in essence, proofreading exonucleases works like a delete key, removing only the most recent errors. ठीक है, तो अभी see एक सवाल इंटरव्यू में ये भी पूछा गया था, like what kind of mutations can get inserted during the replication. तो जिसमें से first ये है, getting the point. There are many, and we are going to learn also. There are many types of mutation which is happening. रेप्लिकेशन के बाद भी होते हैं, ड्यूरिंग रेप्लिकेशन होते हैं, करेक्ट. So this is first answer is that you need to remember is this is how your first mutation can be inserted in the DNA during DNA replication. What is that? That is the addition of the mismatch base pair. And उसका counteract के लिए answer के लिए आपको बोलना है how it can be removed. So that can be done by the same DNA polymerase having the 3' exonuclease activity. Okay? यहाँ तक clear है? Okay.